The problem we will be discussing is why would high-fat feeding cause a reduction in the number of GLUT4 transporters on the muscle membrane?
So the mass that we're going to be looking at is GLUT4 transporters at the membrane into the muscle. The inflow we'll be looking at is the high-fat diet, and the outflow is the change in blood glucose.
Our hypothesis is that exogenous sources of fat further inhibit the insulin signaling cascade via the accumulation of reactive lipid species, ceramides, and DAGs.
With a high-fat diet, we see increased levels of fatty acyl-CoA in the skeletal muscle. When this fat is not utilized for fuel, as is the case in a sedentary individual, it sits and is converted into triacylglyceride and stored in lipid droplets, causing an accumulation of reactive lipid species, including DAGs and ceramides. DAGs are formed through re-esterification. This is when fatty acyl-CoA enters the muscle cell and combines with glycerol 3-phosphate using the GPAT enzyme to form lysophosphatidic acid. This then becomes a DAG when LPAT facilitates the reaction. Through sphingolipid formation, ceramides are made. Both DAGs and ceramides target the insulin signaling pathway and inhibit elements of it. DAGs inhibit IRS, and ceramides inhibit AKT and PKB. This inhibition is problematic. AKT is responsible for relieving the inhibition of AS160 on GLUT4. If AKT doesn't phosphorylate AS160 and release it from the GLUT4 storage vesicle, then GLUT4 will not be transported to the membrane. This is why we believe the mass in our problem, GLUT4, is decreasing.
We found 2 studies that looked at the effect of a high-fat diet-induced accumulation of reactive lipid species in skeletal muscle. One of these studies looked specifically at DAGs, while the other looked at ceramides. The first study we found explored the possible roles of different intracellular fatty acid metabolites such as fatty acyl-CoA, DAGs, ceramides, and triglycerides in mediating fatty acid-induced insulin resistance in skeletal muscle. The study found that an increase in intracellular fatty acyl-CoA and PKC activation were also associated with a significant impairment in insulin-simulated IRS1 tyrosine phosphorylation and IRS1-associated PI3 kinase activity after 5 hours of lipid infusion. The graph shows that when infused with excess lipids, there is more phosphorylation of IRS1 serine. This leads to a decreased phosphorylation of IRS1 tyrosine, leading to decreased activation of the insulin signaling cascade. In conclusion, an increase in plasma fatty acid concentration results in an increase in intracellular fatty acyl-CoA and DAG concentrations. This results in decreased IRS1 tyrosine phosphorylation and decreased activation of IRS1-associated PI3K, resulting in decreased insulin-simulated glucose transport activity. The second study we found determined whether ceramides are increased in muscle from obese insulin-resistant subjects and investigated whether ceramides play a role in the insulin resistance of AKT in human muscle. The results of the study showed a 2-fold increase in ceramides in obese individuals when compared to lean individuals. In conclusion, the decreased AKT phosphorylation could play a role in the reduced ability of insulin to stimulate glucose uptake in skeletal muscle from obese subjects. These studies do support our hypothesis that the accumulation of reactive lipid species, DAGs and ceramides, inhibit the insulin signaling cascade.